I'm a consultant in spinal cord injury at Oswestry. I'm also the president of British Association of Spinal Cord Injury Specialists. Now, I would like to warmly welcome you to our keynote session. I have a great pleasure to introduce Professor Robert Brownstone, a highly accomplished consultant neurosurgeon. Professor Brownstone completed his BSc, MD, and PhD degrees at the University of Manitoba and postdoctoral training in neurophysiology at the University of Copenhagen. After completing neurosurgical training at Manitoba, he practiced in Canada for 20 years as a neurosurgeon scientist. He held the Canada Research Chair in Spinal Cord Circuits. He was appointed as the Brain Research UK Chair and Professor and Head of Neurosurgery at UCL in 2015. He has focused on treating people with movement disorders, intractable pain syndromes, and epilepsy. In his lab, he uses advanced techniques in mouse models to understand motor circuit function and dysfunction to develop new strategies to improve movement and quality of life in people with neurological conditions and injuries. His research is funded from multiple sources, including by Wellcome Trust Investigator Award. He is a fellow of both the Canadian and UK Academies of Medical Sciences. I thank you, Professor Brownstone. Now we look forward to your talk on harnessing neuroplasticity for recovery of motor function following spinal cord injury.
Good morning, everybody, or good afternoon. It's Wednesday morning here, but you will hear this afternoon. Thank you for coming. This is this is the oddest way that I've ever given a talk before, so let's see how it goes. I am going to talk to you about harnessing neuroplasticity for recovery of motor function following spinal cord injury. I am a neurosurgeon scientist at UCL. Who can't advance the slides? For disclosures, I'm not a spinal surgeon. I'm not going to talk about any metal at all. My research hires will be on the next page. I'm a co-founder of a new gene therapy company called Sanya Therapeutics, and I have no financial interest in anything that I'll present today. Turn on my laser pointer. These are the people who have done all the work that I'm going to present. In yellow are the people currently in my lab, a host of postdocs on the left, followed by various alumni who contributed to various aspects of what I'm going to talk to you about. Lab management, technical support listed, and my collaborators on the right. My funders from when I was in Canada are listed on the left, and those here are primarily Wellcome, Brain Research UK, and the Medical Research Council. And I acknowledge my patients for the teaching over many years. They have taught me an awful lot. And in the center here, you see a spinal motor neuron that has been injected with a dye and reconstructed. And you can see how beautiful these cells are. This is from a mouse. So this talk is going to be somewhere on the spectrum. And it's up to you to decide where on the spectrum it is. It could be anything from a complete bore to something that sparks a new idea in you, which is would be great. It could be way out in left field somewhere and not be particularly useful to any of you, or it could sound pretty good but not have any meaning at all. The thesis of this talk is that understanding mechanisms involved in the recovery of motor function after spinal cord injury will lead to the development of strategies to enhance motor recovery. And I'm going to try to get you to that point in the next half an hour. And to understand mechanisms, we need models, and we need to be able to push the boundaries. And everything starts with excellent clinical acumen, as you can see on the right here, and being able to describe the phenomenology of our patients. What is it that they are having trouble with, and what can we learn from them? We take that phenomenology and we can start to use animal models in order to determine the mechanisms for what is going on in their bodies to give them these problems. And I call that, click, next, yeah, the there and back again. So the there takes us from the phenomenology in humans to mechanisms in animal models. And the idea is that we can now take what we've learned and go back to humans in order to help them with their problems. Now, there and back again was the subtitle of the novel The Hobbit by J.R.R. Tolkien. when I first presented this many years ago, people in my lab said, does that mean that we are in Mordor? I hope that that's not the case. Now, I'm not sure what the technical difficulties I'm having are, but hopefully they'll be over. So the objective of this talk is to discuss 5 points of increasing complexity so that I can convince both of you here that the spinal cord contains neural circuits that control complex motor functions. And furthermore, these circuits can mediate recovery of function following spinal cord injury. So why do I study movement? Movement is really the essence of all behavior, and that was said nicely by Charles Sherrington, the Nobel Prize winner from this country about 100 years ago, who incidentally had 3 trainees who also won Nobel Prize. So that is a testament to his greatness. And he said, to move things is all mankind can do, and that for such the sole executant is muscle, whether in whispering a syllable or in felling a forest. But it really wasn't, surprisingly, it really wasn't until 2004 that Kim Anderson, who was then at University of California, Irvine, actually asked people with spinal cord injury, what functions are they missing? What functions, if they could regain one function, what would that be? And while many of us would think, well, maybe that's walking movement. We see people in wheelchairs and surely they'd like to walk. That is actually down the list, somewhere in the middle of the list. And people with paraplegia, the number 1 function is sexual function, of which we know very little about from a mechanistic point of view, then bladder and bowel, trunk stability, and finally walking movement. With people with tetraplegia, it's by far and away arm and hand function that they want. Today, I'm going to talk primarily about walking movement, but I'll also venture into arm and hand function a little bit. Now, what are the consequences of spinal cord injury on movement? Now, you all know that this really depends. And there's quite a difference between those who suffer from degenerative spinal disease and a traumatic spinal disease. And on the left, you have the MRI of someone with degenerative disease who has significant spinal cord compression and yet no myelopathic signs. They're walking fine. They're doing fairly well from that point of view. Versus on the right, you see someone, a young woman, a patient of mine years ago when we did these things called x-rays, who was in a motor vehicle crash that ended up being fatal, as you can see in that picture. Now, the difference is quite obvious, but the difference in movement, we've all seen patients who have significant degenerative disease who are doing surprisingly well. And we think, what does the spinal cord do anyway, given that you don't need much connection from the brain to the spinal cord in order to work? And that's because this has happened over a long period of time, and the spinal cord has homeostatic responses that minimize disability during this slow progression of the disease. So that is, there's neuroplasticity somewhere, and could that neuroplasticity be in the brain or the spinal cord or both? So I'm going to take a minute here to talk about homeostasis and plasticity. Here is a typical general curve that describes homeostasis, where you have your dependent variable and your independent variable, and these dashed lines here describe the region where there is homeostasis. In other words, there can be lots of change in your independent variable with little change in your dependent variable. Now, if we think about this as being circuit function, the function of your neural circuits, and this is the amount of movement, the circuits can be affected to a good degree here, and movement can remain the same. We call that homeostatic plasticity, and that's what occurs during the degenerative spinal disease that I've shown you. Now, we can change where that curve lies. We call that Hebbian plasticity. So exercise will make movement be even better, and disuse will make it be even worse. So that curve can shift up and down. But when we get past these boundaries, or if these boundaries change and the flat part becomes narrower, we come into regions of maladaptive plasticity. So this is plasticity that is not doing any good for the patient. So for instance, in degenerative disease, we have this region of homeostatic plasticity, but over time, we can get at one end flaccidity, depending on where the lesion is, or we can get spasticity, more commonly seen, for example, with myelopathy. So when do these homeostatic mechanisms fail? They fail if the injury is acute, as in that X-ray that I showed you with a spinal cord injury or traumatic brain injury or stroke. They fail before circuits are wired and or neuronal properties aren't developed. So for instance, in developmental conditions, and they fail when there aren't a sufficient number of neurons left. And we see that in neurodegenerative diseases like Parkinson's disease and ALS, but also in Codd-Aquinas syndrome, for example. What does the spinal cord do? So in order to understand plasticity, we have to have some understanding of what the spinal cord does in the first place. And the common thinking is that the spinal cord is a bunch of cables that connect the brain to the muscles. And this is certainly not the case, and I hope to convince you of that. In fact, the spinal cord is more like this. You do have some long cables represented by these long connecting wires here, but you have many integrated circuits within the spinal cord that actually serve some sort of function. So when we think about motor circuits for piano playing, many textbooks and many people will think it looks something like this, which couldn't be farther from the truth, that you have a neuron that some people might call an upper motor neuron in your motor cortex. It has a very long axon that comes down through to the medulla where it crosses over and down into the spinal cord where it innervates your motor neurons and makes your, in this case, your thumb move. And that is not simply oversimplified, that's just plain wrong. So to start with, what is an electric circuit? This is not a circuit, this is a one-way path. And we know, for instance, if I was lucky enough to have a 6-year-old granddaughter and asked her what a circuit is, this is what she would draw. But we know that motor function in the absence of sensory input is non-functional. You need a circuit in order to move. And we know that, for instance, in people with sensory neuropathies, they do not move properly. Secondly, the cortex is far too remote from the site of action and can't do something like control your muscles in patterns such that fingers flex and extend in rapid sequence and play a beautiful sonnet on the piano. So motor circuits are distributed in place and function. And I don't want to minimize what the cerebral cortex does. It is kind of important. And it is important in particular for intention and selection. Intention is more philosophical. I don't know what that is. Selection is selection of the movement of what we want to do. I call those control circuits. Those circuits projected to the brainstem. And yes, some of them project to spinal cord circuits and some even project to motor neurons. But brainstem circuits are responsible for the command. What is the command? How do we take the selection and turn it into a command? That command goes down to spinal cord circuits, and spinal cord circuits are responsible for organization. So spinal cord circuits, for example, ensure that when you want to change a joint angle, you, let's say you contract your biceps and you relax your triceps so that you have control of certain joints. Spinal cord circuits also control the coordination within a limb and the coordination between both limbs so that when you're walking, for example, you don't lift both legs up at the same time. I call those organizational circuits. And finally, those project to spinal muscular circuits. These are your spinal motor neurons that project out to the periphery and innervate your muscles and cause them to contract. And these are all circuits because starting from the outside, you have your limb sensory afferents that project back to 3 levels of these directly in order to modulate their activity. You have auditory inputs coming into the brainstem, visual inputs coming into the cortex, and you have all of these circuits in addition to these downward arrows having upward arrows all communicating with each other. And so you end up with multiple nested loops that produce behavior, and all of these loops are necessary. So the 5 points I'm going to make are going to start with this one. The spinal cord contains circuits that can produce coordinated movement. And so here you have a film from the first half of the 20th century by T. Graham Brown. And what he's done here is he has a decerebrate cat. So this cat has no cerebrum, no cerebral cortex or diencephalon. And this cat is air stepping. And you can see that there's beautiful coordination of the limbs, both the joints within a limb as well as the coordination between limbs, and that is without a brain. And Brown went on to show that you didn't even need the brainstem. And what he did here is he did a spinal transection in the lower thoracic cord in a cat that had no afferents. So he had cut all the dorsal roots going into the lumbar spinal cord. And then he recorded activity in the muscles. And here you have the tibialis anterior muscle and the gastrocnemius muscle below. So a flexor and extensor of the ankle. And you can see that there is beautiful alternating activity, thus showing that the spinal cord itself, with no inputs either from above or from the periphery, could produce locomotor activity. This was nicely described in humans by Lermite a few years later. This is 1919. He described a patient who had a complete spinal cord injury, and there's the histology from after the patient died, of course. And he described spontaneous rhythmic movements of the lower limbs following complete spinal section, again showing that the spinal cord could produce this rhythmic motor output. That was beautifully shown a few years ago by Sir Rossignol and colleagues at the University of Montreal, where they recorded from various trunk and limb muscles on both legs in a patient with a complete spinal cord injury. This is not the patient, just a demonstration of these muscles that were recorded. And what Serge and colleagues showed was beautiful activity that was alternating between the right side and the left side, and alternating between extensor and flexor activity, thus showing that the spinal cord contains the circuits that are capable of producing locomotor-like activity. So that was point one, that the spinal cord has these circuits. Now, what about sensory feedback? Sensory feedback can certainly modulate these spinal motor circuits. And here's another film from Brown of another decerebrate cat on the treadmill. And what Brown is doing here is he's slowly adjusting the speed of the treadmill. And you can see the speed kind of in this arrow at the bottom. And what you can see here is not only does the cat have beautiful coordination, but it responds to the speed of the treadmill. First, the limbs start moving faster. And then as the treadmill speed gets faster and faster, the cat moves from this alternating hind limb gait to more of a gallop. So not only is are the limbs moving faster, but the interlimb coordination changes and responds to the sensory input of the treadmill. So yes, spinal cord circuits can respond to sensory inputs. And so the real question is, how do these sensory inputs affect spinal motor circuits? Can we figure out circuits for sensory motor processing in here? So this is a cross-section of the spinal cord from the cover of a book by Watson et al. Called The Spinal Cord. And here what they've done is they've labeled all neurons with a marker for neurons, so there are no glial cells here. And you can see that the spinal cord gray matter is completely jam-packed full with neurons. We can kind of pick out spinal motor neurons as these large cells in the ventral horn, and we're pretty good at saying that these are motor neurons. We can maybe see a little bit of structure and pain centers in the very dorsal part of the horn, but other than that, there just seems to be a huge mess of neurons. And in fact, one neuron compared to the one beside it might be of a completely different class and a completely different function. So we have a real challenge in trying to pick out motor circuits from these approximately 1 gazillion cells. And the way that we've been doing that over the last 20 years is by using genetic techniques to have markers that can pick out cells within the spinal cord. And so here's a population of cells that I'm going to talk to you before. Here's the cross-section of the spinal cord, the dorsal horn, the ventral horn. We see some motor neurons down here, and we see these green cells that are genetically expressing a fluorescent protein. And these cells are called DI3 interneurons, and we started studying them because they were in between the sensory processing areas of the spinal cord and the motor areas of the spinal cord, and we wanted to figure out how rehabilitative techniques that rely on enforcing sensory input can lead to changes and adaptation in movement. And here, so I don't know whether you see I don't know whether you can see Tuan here. I didn't realize my picture would be there. I can't seem to move it on my screen. But Tuan Bui was a postdoc in the lab for many years who did much of the seminal work on these neurons. And here are the neurons here. So we see the sensory fibers coming in pink. We see the motor neurons here. And I'm going to show you a little bit better here. We've turned off one of the channels so we can nicely see the sensory inputs coming to these cells and the motor neurons there. And this is just about a millimeter of a mouse spinal cord. And you can see that it is jam-packed full of these cells. They're called DI3 interneurons that are in between the sensory inputs and the motor outputs. Next. And when we looked at these neurons, we did many things, and I'll just show you a couple of them. But here we have their one of these neurons here, and we're looking at their sensory inputs labeled with this red stuff called VGLUT1, which is a glutamate transporter that labels all the terminals, and you can see them in this channel here. Here's these two from this box here, and here's a whole bunch from over here. And they have different biochemical profiles. And what that means is that some of them come from muscle afferents and some of them come from skin afferents. So they receive different types of inputs. So we see that they receive multimodal sensory inputs. Here they are in the middle of the spinal cord, and they project to motor neurons and excite motor neurons. And one of the nice things about that is that they contain this glutamate transporter called VGLUT2 that packages glutamate in these sensory terminals and is responsible for communication between these cells. And that allowed us to do a genetic trick where we can eliminate VGLUT2 from these cells. So we don't kill the cells, they're still there, but they can no longer talk to their targets. And when we did that, we wanted to make sure that that worked. So there's a good way to do that. This is Turgai Akai, who did these experiments. So he implanted mice with nerve cuff electrodes on peripheral nerves and recorded from various muscles. And here we're stimulating the sural nerve, so a cutaneous nerve, and recording from a medial gastrocnemius muscle. And here you can see the two shocks as an artifact, and 4 milliseconds later, you see this nice, beautiful reflex that's in the right window for this disynaptic reflex. When we remove DI3 interneurons using that genetic trick, here what we've done is we've given 5 shocks just to really get a reflex, and there's absolutely no response. So these neurons are responsible for that reflex. But I mean, people don't miss reflexes generally. So what does that mean to the mice? And really, this is the test for you, is that one of these mice is a mutant, and you need to be able to figure out which one it is. And I can show you this twice because you may find the answer pretty subtle. But what's happening here is that the mice have normal sensation. They can grip the cage top without a problem. But when there is some slippage in their palms, they can no longer grasp, and they fall into a nice soft bedding there. So I think you can pick up the mutant. And that is the grasp reflex. And we all know the grasp reflex, in particular if we've had kids. I thought this was so much fun. I had 4 of them. And it is a very powerful reflex and probably very important for social bonding. But you can have too much or too little of this grasp reflex, and we've seen that in the news cycles over time. We all know who these people are. That is the Prime Minister of Canada and the President of Prime Minister of Brazil here. So there's too little hand grasp, too much hand grasp, and this was just the right amount of hand grasp back in the 60s. But in neurodegenerative diseases, you can have abnormal grasp. And this is actually quite common in neurodegenerative diseases. And here's a patient of mine who, the harder I pulled my fingers out, the more white that they turned, and I finally had to pry them out, and this was quite disabling for her. And we now are starting to understand the pathophysiology of hand function from those circuits that I've just shown you. So that was point 2, that sensory feedback regulates spinal motor microcircuits. Point 3 is that these circuits are important for plasticity in the spinal cord, and using these circuits can help with recovery of function after spinal cord injury. And here is an example, another cat. This is by Serge Rossignol at the University of Montreal, who has done a complete transection in the thoracic spinal cord of this cat, and then over a 6-week period has trained the cat to walk on the treadmill. And it's got beautiful intra-limb and inter-limb coordination. So that sensory input of being on the treadmill over many weeks has led to plasticity and activation of the spinal locomotor circuits. And that's it can be useful in humans as well. Here's a patient at the University of Alberta, and this is many years ago before we even fully understood treadmill training, who undergoes body weight support treadmill training, and first with 2 therapists here. And then a month or so later, here he's got 1 therapist to correct his footfall. And then he needs no therapists sometime later. And yes, this doesn't translate to perfect walking, but much better walking. And in all quantitative assessments of his walking, his gait is significantly improved and his quality of life is improved. So training and providing that sensory input can lead to adaptation, not only in cats, but in people. And that's not just from the sensory feedback, because sensory feedback is very slow. And we know that from staying in cheap hotel rooms where you turn on the shower in a room that you don't quite know. When the water's too hot, you turn it down, it's too cold, and it takes you a while to oscillate to find the right temperature. That's sensory feedback. And so we need some system other than sensory feedback, and that's a system of what we call comparators. So in other words, anytime you're doing a movement, what you're doing is you're predicting what that sensory input should be. And if that sensory input matches, you're good. But if that sensory input is different from what you predict, you get a reflexive correction of your movement. In other words, you have in control theory, you have a controller that projects to your effectors. Here we're going to call them motor neurons, and that produces an action. You have afferents that come back from your muscles and your skin. Those are sensors and provide feedback to these comparators. But at the same time, the controller has feed forward or predictive input to the comparators, which now compare the predictive input with the instructive input and calculate the sensory prediction error to adjust the controller. And this was nicely described by a famous basketballer, Michael Jordan, who said, "I've missed more than 9,000 shots in my career. I've lost almost 300 games. 26 times I've been trusted to take the game-winning shot and missed. I failed over and over and over again in my life, and that is why I succeed." In other words, we learn from our errors, and here it is at the most basic level. And so finally, point 5 is that the spinal cord contains comparator neurons that are involved in motor learning after spinal cord injury. And so how do we know that? And we know that, for instance, using preparations like this. So this is an isolated mouse spinal cord with various electrodes attached to the dorsal and ventral roots, so we can do various things. In this case, they're attached to the ventral roots. Here's a schematic of that. And the L2 ventral roots contain mainly flexor axons, and the L5 ventral roots contain mainly extensor axons. And so if we record from them and apply some neurochemicals, what we see alternating activity between the right and left flexors. So here you have a flexor burst on the left, followed by one on the right, left, right, left, right, left, right. And if you look at the flexor versus extensor, we see the same thing. This is on the same side. On the right side, we see extension, flexion, and we call that fictive locomotion. So if we go to our DI3 interneurons again, right? So remember that these seem to be acting as comparators in the hand grasp, and all of a sudden the cage is, not all of a sudden, even slowly, the cage is tilted. The cage top is tilted. The mice are not processing that change in sensory response properly. So here we record from one of these DI3 interneurons, and we see that it is active during extension only. So in white, we have the extensor phases, and gray, the flexor phases, and you can see it's always quiet during flexion. These are action potentials. So really what we have with these DI3 interneurons is we have various sensory modalities from the skin and from the muscles coming in and forming synapses with these DI3 interneurons that project to motor neurons. And we have the spinal locomotor circuits that project to these DI3 interneurons as well. So they can now compare the motor commands with the sensory inputs, and if there are any problems, they can make adjustments to your motor output. And so if we eliminate them, what does their walking look like? Well, their walking actually looks pretty good. If you've seen enough of these, you can detect the difference, and you might be able to anyway, that the one without the DI3 neurons has feet that are slightly wider apart than the ones above. But overall, they are walking pretty well. They do very well without their DI3 interneurons. But what happens if you cut their spinal cords and then train them to walk on a treadmill? Here's what a normal mouse will do. You see that on the left. And yes, the walking isn't perfect, but they can move their feet, and you can see actually pretty good foot placement just before, I'm going to play that again, just before the foot falls on the treadmill. And here you see one without these comparator neurons, without the DI3 neurons that can't learn to walk. I'm showing this at slow motion so that you can see it. There's Nico Stefani who did these experiments in my lab. So as comparator neurons, DI3 interneurons integrate the predicted and the actual sensory activity in order to adapt and to learn motor behavior. So the big question is, can we target them to improve neurorehabilitative strategies? And here they are. Here are 3 mice again, and I think you can see which ones have their DI3 interneurons intact. They all have complete spinal cord injuries, and you can see the 2 on the top move their hind legs that help them propel themselves into their little homes. But the one on the bottom is doing so only with their forelimbs above the level of the spinal cord injury. So DI3 interneurons are key to spinal motor learning after injury. Here is the supraspinal centers that are now interrupted with complete transection in the thoracic cord coming down. And here's the spinal cord with locomotor circuits that are telling motor neurons what to do, as well as telling motor DI3 interneurons, comparator neurons, what sensory input to expect. Here's the sensory input coming in. And we have, after transection, we have recovery of locomotor function. On the other hand, if you don't have these DI3 interneurons, remember all we've done is genetically remove them from the circuit, but what we have is absence of any recovery of function. So despite the acute injury of spinal transection, there's normally some sort of homeostatic process that you can induce with sensory inputs such as treadmill training. Recovery is incomplete because we don't have connectivity to higher centers that can modulate this. And when the local circuit substrate is also disrupted, then there's going to be no recovery of function. So I'll close there, hopefully on time, to summarize that the spinal cord contains circuits that can produce coordinated movement, and sensory feedback can regulate these circuits. Sensory feedback can induce plastic changes in the spinal cord, and is necessary but not sufficient for motor learning. You need the circuits in the spinal cord as well. And ultimately, you want to be able to modulate these circuits from commands from above, which is a completely other topic. The spinal cord contains comparators that compute the difference between the predicted motor commands and the actual motor outputs. So these are involved in motor learning after spinal cord injury and could be possible targets for interventional neurorehabilitation. I'd like to just close by saying that understanding mechanisms involved in recovery of motor function after spinal cord injury will lead to the development of strategies to enhance motor recovery. We can target these cells genetically. We can learn how to do that and hopefully improve neurorehabilitative strategies. And with that, I thank you very much.
Naveen, unfortunately, you're muted.
Thank you very much, Professor Brownstone. There is exciting information and data. From the service user's perspective, recovery of motor function following spinal cord injury is very useful, but recovery of bladder and bowel control is preferred. In the future, is there a role of exploring circuit function in improving and restoring these functions?
That's an excellent question. I just want to make sure I'm not on mute. Can you hear me?
Yes, we can. Yeah.
Yes. Okay, great. Excellent. So one of the issues is that we've been studying walking, as I've shown you, for well over 100 years, and we have a very good understanding of the circuits involved in walking. we have much less understanding of control of our sphincters. We have some of our urinary sphincter, but less of our anal sphincter. So we need to understand the physiology of those muscles a little bit more. I think that there is hope. I think it's just that it's farther behind where we are with walking movements and with hand function. You're still on mute.
I can't see any questions or comments from the chat. So I would like to thank you, Professor Brownstone, for accepting our invitation and for delivering your keynote lecture. Thank you very much.
Opportunity.
Thank you.
Okay.
So it looks like the veins are popped off. Has anyone got any questions for Professor Brownstone at all? Please do put them in the chat. I think that your talk was very informative and has stung a lot of people into complete method.
It's an interesting way to do things, remote. This remote lecturing and everything, it's different. Okay, I see two questions there. Nigel, you're asking me, do you think there would be more potential in the immature spine? That's a fantastic question, but the question is, how immature is immature? And certainly we know that the younger you go, the better that rehabilitation is in general for any spinal cord or brain injury. And so, yes, I think the younger the spinal cord, the more chance of rehabilitation and the more chance that we have to intervene. In fact, if you go to fetal spinal cords, they will regenerate. So if you go way back, you can get excellent recovery of function. Any comment on the role of stem cells to spinal cord injury? I have lots of comments on stem cells and spinal cord injury. I didn't talk about them today. Actually, it's my one paper that I've done with the philosophers on the ethics of using stem cells for spinal cord injury. That was in 2011 in Cell Stem Cell, if you really want to see what my opinion is. But I think that the issue with stem cells, and I do work with stem cells as well, is that you need to have very specific goals of what you want to do with the stem cells. So for instance, do you want them to provide a trophic factor? Do you want them to remyelinate axons? Do you want them to replace neurons? Do you want them to act as relay neurons? And many of the stem cell studies are just, wow, we can make stem cells. Stem cells are magic, yet let's implant them into the spinal cord and they will do their magic and fix spinal cord injury. So I think that we are a long way away from using stem cells to repair spinal cord injury. But again, you never know. You never know when that moment is going to come where somebody in some lab has a terrific discovery that is going to be translatable. I think we have to be particularly careful with this population of patients. One of the points there is that usually these are young men with spinal cord injury. They will have heard of stem cells. They present with an acute spinal cord injury, and you say, well, we can enter you in the study and inject some stem cells. The tendency is going to be for them to say yes. So we have to be particularly careful from an ethical point of view that that these studies are done properly as studies with proper informed consent. Next question. For humans, what is it? It keeps moving. What is the time frame for the rehab in walking? So I think that that already we are doing better with rehabilitation for walking. I think that body weight support treadmill training has been terrific, and now we have the addition in some centers of epidural spinal cord stimulation that can certainly facilitate the training. So I think that I think that we are getting there. Certainly from a research perspective, we've done very well. I think there's a ways to go to translate that into clinical use. We still need to understand what we're doing with the spinal cord stimulation and how that interacts with rehabilitation in order to optimize that. These are very expensive and time intensive, but that shouldn't stop us from pursuing that. I do want to remind you that walking is on the list, but it's way down the list of things that we need to treat. Is there a time period where the neuroplasticity lasts? For example, could we look at training a few years after injury, harnessing neuroplasticity? Yeah, so that's a great question, is what is the window that we need for this? So again, with spinal cord injury and treadmill training, they have people who have used these studies have gone several years after the injury. It seems that the earlier the better, at least to a point. But I think that that's something that we still need to learn. And we're learning from our colleagues who study stroke and other neurological injuries, acute injuries, about these time windows, as well as about the doses of training that are needed. Is there a point of no return from which treadmill walking would be pointless? I think that's the same question. One of the issues with walking that I didn't mention is that wheelchair technology is actually really good. And you compare that to other problems that people have, like upper extremity dysfunction, where we don't have very good prostheses. Bladder and bowel, we don't have very good solutions. So that is something to keep in mind when thinking about walking. Whoa. This is not really moving quick.
Professor Brownstone, thank you very much for that. We are almost out of time, and thank you very much for reading some of the questions. There are lots of questions that are still coming in. So may I suggest that once this session is over, we can obviously send you the questions. I'll also encourage all attendees to go into the networking lounge into the chat function, and maybe our questions can be answered there after the session. So we are almost coming to the end of this. So I'd just like to thank you, Professor Brownstone, for your talk, and also for your questions. Unfortunately, I'm not too sure where they've been gone to, but thank you very much for that.
It's my pleasure. Thanks for the opportunity.